Hello everyone. First of all, welcome to Bioenigma Uncovered. Today we are going to begin with one of the most important skills that every bioinformatician, computational biologist and genomics researcher must learn. And that skill is Linux. Now many of you may be hearing about Linux for the first time or perhaps this is your first time using it. So there is no need to worry. Today's session is completely beginner friendly. Let me ask you a quick question. How many of you have heard about Windows and how many of you have heard of Linux? By the end of today's session, you'll understand what Linux is, why Linux is essential in bioinformatics, what the Linux terminal is, and how basic Linux command works. And most importantly, today's goal is not to memorize commands. Today's goal is simply to become familiar with Linux. Now, why Linux in bioinformatics? Now, the question is, if we are already using Windows, why do we need to learn Linux? Imagine, tomorrow you are asked to analyze NGS data, microbiome data, RNA sequence data, whole genome sequencing data. The tools you will use such as FASTQ BWA, Bowtie2, SAM tools, Chime2, all work much more efficiently in Linux environment. Linux is standard operating system used in research laboratories, universities and high performance computing clusters worldwide. That's why Linux is often considered the language of bioinformatics. Just as reading research papers become difficult without knowing English, performing bioinformatics becomes difficult without learning Linux. Imagine you have a folder containing 5,000 FASTQ files. Would you manually open each file? Of course not. Instead, Linux lets you type, find, name, FASTQ. One command and done. There's another example. Imagine you have 10,000 patient files and you need to find all files containing the TP53 mutation. On Windows, this could take hours. On Linux, it's just grep T53 and here we go. Just a few seconds and we will find it. Now what is Linux? In simple terms, Linux is an operating system just like Windows, Mac OS, Linux. All of these three are operating systems. The main difference is that Windows relies mostly on the mouse and graphical interference, whereas Linux allows us to perform most tasks using commands. And that's exactly what makes Linux so powerful. Window you are seeing is called the terminal. In bioinformatics, you will spend a lot of time working inside this terminal. At first, it may look intimidating, but trust me, after just 2 or 3 sessions, it will become your best friend. Now, you might see something like this, base, chahat, @ $chahat, $. This may look confusing at first, so let's break it down. Chahat with small c, it is a username, and Chahat with the capital C, it is the computer name, and the $, it indicates the Linux is ready to accept your commands. Now, the first command is to know your location. So let's begin with our first command, pwd output mntd. So pwd stands for print working directory. In simple words, it tells you where you currently are. Just like Google Maps, it shows you current location. Linux uses pwd command to show your current folder. In simple words, it tells you where you currently are, just like Google Maps. Now, next command is ls. It is listing files. Now type ls. ls stands for list. It displays all the files and folders inside the current directory. As you in NCBI, I am just opening the site first. Then I'll be going from all databases. I'll be selecting the gene, and in the search box I'll be typing SLC12A3 gene, that is a solute carrier gene in humans. Then I am going to the gene sequence. I have selected the gene, and then I am downloading the FASTA file of the same. I have downloaded it and to that for specific folder and checked the list with it, and it has the gene.fna. Now if I want to check the whole sequence, and I won't be opening the whole file, I'll be just typing head gene.fna. That indicates the first few lines of the same file. And similarly, tail sample.fastq I have typed. The tail command shows the last few lines of that specific file I have downloaded from NCBI. Imagine your FASTQ file contains 5 million sequencing reads, and you open the entire file? No. Instead, you will be using head command or tail command. Next command is copying file. To copy a file, cp command is used. For instance, I have typed cp sample.txt, and the output also sample underscore copy.txt. So my file is copied when I typed ls again, and I can view my file. Next command is deleting files. To delete a file, rmsample.txt. Rm command is used, that means remove. A very important warning, unlike Windows, Linux usually doesn't have a recycle bin. Once deleted, the file is permanently removed. So always use the rm command carefully. Next command is for searching a file. Suppose your project contains hundreds of files and you only want to locate FASTQ file. Use find name FASTQ. This is one of the most useful command in NGS analysis. Next command is for counting lines. wc-l sample.txt. This counts the total number of lines in the file. Here's an important bioinformatics fact that every sequence read in a FASTQ file occupies 4 lines. So number of reads is equals to total lines divided by 4. As I have checked the number of lines in this specific file I have downloaded from NCBI, and it contains 1450 files in gene.fna of SLC12A3 gene. Now, let me ask you another question. Do you think Linux command is only in bioinformatics? Most students use. Now, let me ask you another question. Do you think Linux is used only in bioinformatics? Most students usually say yes. But the answer is no. Linux is everywhere. Android phones. How many of you use an Android phone? Android is actually built in Linux kernel. Next is Google. Thousands of Google servers run Linux whenever or you search on Google, your phone goes to Google's Linux server and search results. Another is YouTube. Millions of videos are streamed every minute, and behind the scenes Linux servers makes it all possible. Another Linux example of CD stands for change directory. Now, for instance, I have moved to cdmntt, that is that I am in D directory, D drive of my PC, and here I want to move to folder named Chahat. So I typed cd Chahat. So I moved to Chahat folder, and in the Chahat folder I have Bioinformatics folder. Similarly, I have typed cd Bioinformatics. So I am in cd Bioinformatics folders, and then I typed ls, which shows the list of the files which are already present inside the same folder. The next command is detailed view command. If you want a more detailed view of the files and folders, you can use this command like ls-l, whichever command appears Now, this will give the detailed view of the files, like when these files were created, at what time were created, how much of data it contains. Everything will be viewed in details. Next is creating a folder. Let's say we are starting a new bioinformatics project and I'll be typing the mkdir test. So mkdir stands for make directory. A directory simply means a folder. Now let's run ls and we can see the new folder has been created. Now next is creating files. Folder contain files. Now create a file. I have written touch sample.txt. I want a txt file. So here I typed ls again. Now the test file is appeared. Sample.txt. Next is viewing the content. Now let's write something inside the file, that is echo hello bioinformatics sample.txt. I have written it in the sample.txt file. Now if I want to view it, I'll be typing cat command. I typed cat sample.txt. It shows hello bioinformatics. As you can see that I typed with the echo command and I can view my file content with. Let me ask you another question. Do you think researchers perform every step using a mouse? No, Linux automates everything. Now another important question is, why do we perform genomics, transcriptomics and metagenomics? Is it simply to publish research papers? Absolutely not. The ultimate goal is to improve patient care. Suppose genome analysis identifies a disease associated mutation that helps us to understand the disease mechanism. Next, we can identify biomarkers for early diagnosis. Next, we can identify biomarkers for early diagnosis. If we discover a protein or pathway involved in disease, it may become the drug target. Pharmaceutical companies can then develop therapies against that target. Today, personalized medicine is also possible because of genomics. Instead of giving every patient the same treatment, therapy can be tailored according to an individual's genetic profile. Similarly, metagenomics help us identify disease-associated microbes, opening the door of microbiome-based therapeutics. During the COVID-19 pandemic, bioinformatics played a major role in vaccine development. Similarly, metagenomics help us identify disease-associated microbes, opening the door of microbiome-based therapeutics and next-generation probiotics. During the COVID-19 pandemic, bioinformatics played a major role in vaccine development and variant tracking. So the final goal of the Linux, bioinformatics and omics technologies is not to data analyze. The ultimate goal is better diagnostics, better therapeutics, and better patient outcome. So if you have any questions, feel free to ask in the comment section. You might be wondering why we are learning these commands. Is bioinformatics only about creating folders or viewing files? Absolutely not. The commands you have learned today from the foundation of bioinformatics. Like imagine I have received a sequencing dataset containing 10 million reads using Linux. I will locate the file, check its size, preview the first few reads, copy it, move it into my analysis directory, finally process it using tools like FASTQ, BWA, Chime2 or many others. Everything we have learned today will be applied directly to real sequencing data in future sessions. Thank you and I'll see you in the next lecture.